Hello and welcome back to our second video on energy, and specifically this time we're going to be looking at enzymes. So enzymes are proteins, and if you remember back a couple weeks ago we talked about them in terms of the biomolecules. So they are polymers made up of amino acids, but specifically we're going to be looking at the proteins that help metabolism in the human body. So enzymes are proteins that enable chemical reactions in our body. So enzymes are catalysts. And what that means is that they speed up chemical reactions. And the enzymes that we're going to be talking about are all proteins, and we'll kind of go from there. So we're going to be looking at proteins that speed up chemical reactions inside the human body. And here's the model that we're going to look at. And there's a few terms here: enzyme, substrate, and active site that you've got to be familiar with. And so this is something that we're going to look at, and we're going to expand a little bit later, called the lock and key model. But if you look at this diagram, starting over on the left, we've got the enzyme right here in the blue. And on that enzyme, it has what's called an active site. And this is a specific shape. Remember, a protein is this folded structure, and this active site has a specific shape that will fit only one substrate. And they fit together just like a lock and key, and they form what's known as the enzyme substrate complex. And that enzyme holds the substrate in place and makes it easier for a chemical reaction to take place that will, in this case, split the substrate into two pieces, and we get our new products. So this might be like the digestion of sucrose, which is two monosaccharides into the individual monosaccharides there. So the terms that you really need to be familiar with, once again, are enzyme, the active site, the substrate, and then the products. So the enzyme is the actual protein. The substrate, or substrates, are the reactants which bind to enzymes and are converted to the product. The active site is the area on the enzyme that binds the substrate and holds it. So here is a model that's a little bit closer to what the actual protein might look like. So if we start over here on the left, here is kind of what they call the ribbon model, which shows the backbone of those amino acids that form the protein. And here's the active site right here. There's a little cleft that the substrate would fit right into. And if we kind of look at the ball and stick, or the space fill model, we can see the same thing. We've got an active site, and that's where the substrate fits right in, kind of like your hand into a glove. It's called the lock and key model. And when it binds, it actually pinches together a little bit tighter into something what they call the induced fit hypothesis, which says that the enzyme will actually change its shape. A conformational change takes place, and it squeezes that substrate even tighter and makes the fit even better. And here's a great, if you look at the website down below, here's a great website that we're going to kind of go through. I'm going to take you to just for a couple minutes so that we can see how this actually works. And I think this is a pretty good example of the website and how the enzyme substrate complex will form. So here we go. So an enzyme is a protein found in all living things that help catalyze or speed up certain biochemical reactions. Certain types of enzymes will cause a reaction with certain types of molecules, which are called substrates. We talked about that, how they fit together. Every enzyme has an active site which allows it to fit together with a substrate like a lock and key. When the two bind together, the reaction may cause the substrate to break apart into smaller pieces called products. So we talked about the digestion of sucrose into glucose and fructose. When the two bind together, the reaction may cause the substrate to break apart into smaller pieces called products. When the two bind together, the reaction may, and so it's repeating the same thing, but it's going through, and we can see that there's a split. So the enzyme holds that substrate in place, and it makes this reaction easier to take place. And when the two bind together, the reaction may cause them to break apart. And here are our two new products that have now been released. For example, an amylase is a type of enzyme that breaks down starch or glycerin. So in this case, the starch would be the substrate. And in nature, bacteria and fungi secrete amylases to aid in digestion. So in our gut, in our intestine, we have these bacteria that will help us break down these starches. And the enzyme breaks down the starch into soluble products such as glucose, which can then be absorbed into the cell. So we're trying to break it down into that monomer that we can use for energy. The enzyme breaks down the starch into soluble products, and that can be absorbed. So here's our individual glucose molecule that is easier for us to use as energy. And then we're back to the beginning. So now we'll go back to our presentation and kind of move forward with that. So what we need to know about enzymes is how they work. We know that we've talked about them as being catalysts, so that means they speed up reactions. But how do they do that? And what they do is they lower something called the activation energy. So the activation energy is the energy needed to start a chemical reaction. And if you look at the graph, we've got the high point up here. And without an enzyme, this is the amount of energy that we would need to add to get that reaction going. It's kind of like rolling a boulder down a mountain. You need a big push to get it going, but once it goes on its own. Or sliding. You need a little push to get the sled going. That's the activation energy. Now, what enzymes do is right here, we can see, now I'm going to use blue, is it lowers the activation energy. It lowers the amount of push that we need to get that reaction starting so it can happen a lot quicker. So they act as catalysts by lowering the activation energy and allowing these reactions to start and proceed a lot faster. So here's one that kind of shows the activation energy as a hurdle. So we can see without an enzyme, there's a very high hurdle to get over, and not many of the substrates are going to be able to do that. But with the enzyme, it lowers the activation energy needed and allows us to get to the product much more quickly, and many more reactions will result in the production of that product. So we talked a little bit about it, but here it is. So what it is, the enzyme substrate complex works by something called the lock and key model. So the active site and the substrate or substrates have complementary shapes. They fit together kind of like your hand in a glove. And the key thing here is they're very specific. For the most part, it's one enzyme, one substrate. They don't work for more than one. So this next diagram that we're going to look at right here shows very well with the red enzyme. It has a very specific shape of its active site, and the substrate, the yellow piece that fits in there, matches perfectly. That's the lock and key hypothesis. And by holding that substrate in place, it allows us to make the two products over here that are the end of the reaction. Now, another thing about enzymes is they are not changed. They're not changed by the reaction. This means that this enzyme is now free to go and grab another substrate and carry out the same reaction hundreds or thousands of times in the body. And that wraps up our kind of introduction to enzymes. And so here we go. We've got a steer talking to a cow. Says, it has nothing to do with you, Bessie. It's just that I'm lactose intolerant. And for people who are lactose intolerant, that means they lack the enzyme lactase that allows them to break down the sugar found in milk. So again, thank you for your time, and we will talk with you soon.